Extracorporal growth of the inner ear takes place in a plastic dish. The outer rim of the dish is moistened with sterile distilled water. The inner well is filled with 1/10 of a milliliter of tissue culture fluid. The tissue culture fluid consists of Newman and Titel's culture media supplemented with 20% fetal calf serum, 1% glutamine, and antibiotics. The dish is sealed with sterile Vaseline and placed in an incubator at 34.5 degrees centigrade in a 5% carbon dioxide atmosphere. A normal CBAJ mouse bred with a normal C57 mouse is used. At the 12th day of gestation, the gravid mouse is sacrificed by cervical dislocation. The abdomen is washed with 95% ethanol and opened. The gravid uterus containing 7 embryos is removed. The uterus containing the 7 embryos is placed in a petri dish containing pre-warm Newman and Titel's culture media. The embryo is removed from the uterus with 2 pre-sharpened number 5 Dumont watchmaker's forceps. The otocyst is located on the ventral posterior portion of the rhombencephalon above the embryonic carotid artery. The pointer shows the position of the otocyst. The otocyst is now removed from the embryo. The free otocyst is further dissected to remove the adherent mesenchyme and rhombencephalon. The isolated otocyst is placed in the previously prepared organ culture dish which contains the warmed organ culture media. The dish containing the otocyst is closed and returned to the incubator. The otocyst remains in the dish for 5 to 10 days to undergo the extracorporal growth of the inner ear. The 12-day otocyst, which has been in culture for 12 hours, consists of a dorsal portion, a ventral portion, the statoacoustic ganglion complex, and the endolymphatic projection. The otocyst is surrounded by mesenchyme. This sequence is an actual time-lapse motion picture of the growth of a 12-day otocyst for the next 6 days in the organ culture dish. The 12-day otocyst, when removed from the embryo, consists of the dorsal portion, ventral portion, the statoacoustic ganglion complex, and the endolymphatic projection. During these first 2 days, changes in the shape of the otocyst become apparent. At the end of 2 days in organ culture, a number of structures can be noted in the developing inner ear. These are the posterior semicircular canal, posterior ampulla, lateral semicircular canal, saccule, cochlea, statoacoustic ganglion, utricle, anterior ampulla, lateral ampulla, and the anterior semicircular canal. This time-lapse sequence records the 2nd to 3rd day of extracorporal growth of the inner ear. There is a further development of the posterior semicircular canal and posterior ampulla. The lateral semicircular canal and the saccule become more apparent. The cochlea elongates and changes its shape. The statoacoustic ganglion complex begins to separate into scarpus ganglion and the spiral ganglion. The lateral and anterior ampullae become more evident. At the end of 3 days, all of the organelles of the inner ear can be more clearly seen. These include the posterior semicircular canal, lateral semicircular canal, posterior ampulla, saccule, cochlea, spiral ganglion, Scarpa's ganglion, utricle, lateral ampulla, anterior ampulla, and the anterior semicircular canal. Development now continues during the 4th day of growth and culture. Development of the crus commune is seen at 12 o'clock. The posterior ampulla takes on more of its mature shape. The cochlea expands and the apex begins to coil. The spiral ganglion cells begin to migrate into the concavity of the cochlea. Cartilage has begun to form on the periphery of the specimen in the lower left. At 3 o'clock near the base of the cochlea and in the area of the anterior ampulla and anterior semicircular canal. At the end of the 4th day, further progression of growth is seen in the posterior semicircular canal, posterior ampulla, lateral semicircular canal, saccule, coiling cochlea, spiral ganglion, Scarpa's ganglion, utricle, lateral ampulla, anterior ampulla, cartilage, and the anterior semicircular canal. This sequence records the growth during the 5th day. The cartilaginous capsule is becoming more evident. The space between the cartilaginous capsule at 9 o'clock may be the beginning of the oval window. The cochlea is increasing in size and changing its shape. The spiral ganglion and Scarpa's ganglion are now almost totally separated. At the end of the 5th day in culture, the otocyst has developed into a complete inner ear. The posterior semicircular canal, posterior ampulla, lateral semicircular canal, saccule, cartilaginous capsule, cochlea, spiral ganglion, Scarpa's ganglion, utricle, lateral ampulla, anterior ampulla, and anterior semicircular canal are all clearly recognizable. This sequence records the growth and culture from the 5th to the beginning of the 7th day. At the end of this sequence, the otocyst has undergone development comparable to a stage 1 or 2 days before birth. The cartilaginous capsule has completely surrounded the membranous labyrinth. The cochlea has coiled. The 2 ganglions have completely separated. The semicircular canals are distinct. The ampullae appear as dark thickenings in the canal walls. The utricle and saccule have developed. These time-lapse studies have shown the extracorporal growth of the 12-day otocyst for 7 days. The inner ear has developed from a simple sac consisting of a dorsal portion, a ventral portion, a statoacoustic ganglion complex, and an endolymphatic projection into a complete membranous labyrinth with a surrounding cartilaginous capsule. In cultures which have been grown for 10 days, the final stage is comparable to what the inner ear of the mouse would be like at birth. This is a photomicrograph of the histology of the dorsal portion of the 12-day otocyst. The upper smaller circle is the endolymphatic projection. The lower larger circle is a cross-section of the dorsal portion of the otocyst. The endolymphatic lumen is surrounded by epithelium. The pseudostratified columnar epithelium in the lower portion of the otocyst is the area which will produce the cells of the vestibular labyrinth. This photomicrograph shows the ventral portion of the 12-day otocyst stained with hematoxin and eosin. The endolymphatic wall consists of pseudostratified columnar epithelium, which will give rise to the cells of the inner ear. The dense mass of cells to the right is the undifferentiated statoacoustic ganglion complex. This and the next 4 photomicrographs are sections taken from inner ears which were grown from 12-day otocysts. They were all kept in the culture dish for 9 to 10 days. The cultures were then fixed, sectioned, and stained with protargol. This is a crista with well-formed sensory hairs, ciliary tufts, and a cupula. This is the ampulla of a lateral semicircular canal. The well-differentiated sensory cells with ciliary tufts are seen. The cupula extends from the crista to the roof of the ampullary wall. Both the endolymphatic and perilymphatic spaces are seen. Shown here is a macula with well-developed sensory and supporting cells with ciliary tufts. There's also development of an otolithic membrane. The endolymphatic and perilymphatic spaces can be seen. This micrograph shows the anatomical relationship of the cochlea in the middle of the field to the macula of the saccule in the lower left. The cochlea can be recognized by the formation of the limbus, tectorial membrane, and the hair cells. This is comparable to the histologic picture of the mouse cochlea at birth. A high magnification of the organ of Corti shows, beginning at the left, a row of large round cells which are the young outer hair cells. The light area is the early tunnel of Corti. The nuclei of the differentiating pillar cells are at the bottom of the tunnel on the immature basilar membrane. The next 3 large round cells are the young inner hair cells. The limbus, consisting of epithelial and stromal cells, is seen on the right. The amorphous substance above the limbus is consistent with the histological picture of the tectorial membrane at birth. There are more than 30 genetically deaf strains of mice. The first strains were called waltzing mice because of their circulating behavior. This behavioral characteristic is due to the lack of a functioning vestibular apparatus. The homozygotic Chrysler mouse on the left demonstrates this characteristic. Additionally, this mouse is deaf. The normal mouse is on the right. The next time-lapse sequence will be that of the Chrysler otocyst. On the left is an affected homozygotic Chrysler otocyst, and on the right, a normal heterozygotic littermate. The otocysts were removed at 12 days of gestation, and the cultures will be followed for 4.5 days. The 2 otocysts are grown in the same dish. On the left, the gene action of the homozygote is demonstrated by the lack of formation of a normal inner ear in a healthy culture. On the right, the otocyst, which carries only one Chrysler gene, the heterozygote, develops normally. The semicircular canals, ampullae, saccule, and cochlea are all apparent. The growth of the cochlea is especially prominent in the right-hand specimen, the non-affected heterozygote. The affected homozygotic Chrysler otocyst on the left has very little differentiation. In the dorsal portion of the homozygote, a small protrusion can be seen which represents an abnormal beginning of a cochlear duct. There are no semicircular canals. The inner ear on the left would result in deafness and an absent vestibular response. If it were in a mouse, the animal would circle and not respond to sound. The organ culture system demonstrates that at 12 days gestation, the cells of the inner ear of the homozygotic Chrysler have already been genetically abnormally programmed. The following time-lapse sequence shows the early commitment of cells of the undifferentiated otocyst to become specific portions of the inner ear. The otocyst has been cut in half. Each half was placed in the organ culture dish. On the left is the dorsal half of a 12-day otocyst, and on the right, the ventral half of a 12-day otocyst. Their growth is recorded for 5 days. The dorsal portion of the otocyst on the left is differentiating into 3 semicircular canals and their ampullae. This will come about without the statoacoustic ganglion. The ventral half of the otocyst is differentiating into the cochlea. The cochlear duct can be seen forming in the upper portion of the specimen and coiling towards the left. The right-hand specimen contains the statoacoustic ganglion. Other experiments have shown that the utricle and saccule arise from the middle of the otocyst. A series of experiments have shown that as early as the 11th day of gestation, each portion of the otocyst has already been programmed to form specific sensory structures of the inner ear. At the end of 5 days of growth, there is on the left a labyrinth consisting of only 3 semicircular canals and their ampullae. On the right, there is a labyrinth which contains only a cochlea and its ganglia. This study is that of a normal 12-day otocyst whose growth is followed for 5 days. The 12-day otocyst consists of the dorsal half, the ventral half, the statoacoustic ganglion complex, and the endolymphatic projection. The undifferentiated otocyst